Hello everyone, my name is Lily, and today I'm going to show you guys a little bit of the biological components contributing to social anxiety disorder. Starting off with what is social anxiety disorder, it's also known as a phobia or social phobia and referred to as SAD. It is characterized by persistent fear of one or more social or performance situations in which the person is exposed to unfamiliar people or to possible scrutiny by others. Examples of fears can be public speaking and performing in public. SAD interferes with daily life, such as any anxiety disorder does, and people tend to avoid the places that they associate with anxious memories. Moving on to the next slide, these are the questions I will be discussing. One about serotonin's role in SAD, amygdala's connection with memory formation and fear responses, and then any heritability with SAD genes. Right. So then starting with serotonin, a PET imaging study was conducted to measure the role of serotonin. One test measured the synthesis rates of serotonin, and then the other looked at how much of the SERT transporter was involved. The SERT transporter reuptakes serotonin back into the presynaptic cell. An elevated SERT gets serotonin out of the synapse faster, and it could be overproducing the serotonin. The study found a strong relationship between the amount of serotonin produced and the amount of transporters involved. So high serotonin levels and high SERT in the regions of the brain that regulate fear is a significant aspect of SAD formation. An example we learned in class with another serotonin reuptake was when Prozac partially blocks the normal reuptake of serotonin, so the extra serotonin is in the synapse and its effect with positive moods is enhanced. In this situation, the serotonin reuptake is not blocked, but instead is making and ridding of serotonin at a rapid speed. Right, I'm talking really fast, I'm sorry. So now we move to the amygdala's connections with other brain areas that form SAD-type memories. Social anxiety includes memories of social settings that are hard to erase and strong memories in their past experiences. The study used fMRI to measure the brain at rest before and after the fear application, and then they used classical fear conditioning with a conditioned, neutral, and unconditioned stimulus. There was a connection found with amygdala and dorsal anterior cingulate cortex. The dorsal cortex monitors cognition and motor control, and it makes the memory overwhelming emotionally. There was a decreased connection with the amygdala and the medial prefrontal cortex, which regulates social perception, memory, and emotion. The medial prefrontal cortex lowers amygdala influence when paired, so the controlling of emotions are not as successful and hard to get rid of. Then we have increased insula and hippocampus connection, and the insula is in charge of pain and temperature, emotional awareness, and decision making. This pairing makes a memory more specific and easily activated. This all being said, memories involving fear and anxious experiences are more prevalent when the amygdala is paired with the other brain areas that control emotional expression. In class, we learned about the limbic system, which is made up of the hippocampus and amygdala. The hippocampus has memory formation, and the amygdala is all about emotion. Both are near the fornix and thalamus, and also the frontal lobe as a whole is responsible for decision-making, impulse control, judgment, and emotional control. Lastly, we are going to cover heritability with SAD. A genome-wide association analysis was done to determine the SNP-based heritability of social anxiety. SNP stands for single nucleotide polymorphism, a little bit of a mouthful, and it refers to a single letter variation in a person's DNA. What is being measured is how much trait variation is explained by the combining of SNPs. These are common variants. So for example, one person might have an A and another a T. So the SNP heritability estimates about 12%, and these are with the common gene variants contributing. SAD is polygenic, so as we learned in class, it is when multiple genes play a role and each have distinct effects with a trait. Now with the twin study, it's a different study, it was conducted to measure the vulnerability to SAD. The risk factors for SAD are heritable with around 66%, so the genes are huge when it comes to vulnerability, but different environments also play a role with how much vulnerability is still present over time. This does not agree with genetic determinism, which is when genes play the primary role in our genetics, but as we learned in class with the Diasis, I think I'm butchering that, Diasis stress model does tie in. So the Diasis stress model supports that initially vulnerability is genetic, but environmental stressors later on in life determine the likelihood of someone developing a disorder or trait. That being said, genetics are important when diagnosing SAD, but environmental factors are also noted as crucial. Sorry. That concludes my presentation. There is still much to learn about SAD, but there is a lot to say about our fear regions, like the amygdala playing a huge role, serotonin being one of the most used neurotransmitters, and the contribution of genetic and environmental factors affecting the likelihood of developing SAD. I hope this wasn't too boring, but I thank you guys for whoever's watching this and listening. Oh, and there's the bibliography, but we don't need to see that. Bye!